Good afternoon and welcome to the sixth in our 2021 webinar series delivered by Dr. Tim Sandle. I hope you didn't miss us too much last month, but we're here now until the end of the year. These monthly webinars are covering different aspects of Annex 1. Hopefully they will help you prepare for when it goes live later. Today's topic will cover spores in my clean room, remediation and disinfection strategies. Before we start, just a couple of housekeeping issues to think of. If you have any technical issues, if you can send a short message in the questions box on the right-hand side of your screen, we will try and answer them as much as we can. Please be aware that we're working remotely, so it's not always easy for us to resolve your issues. During the webinar, we'd also like to invite you to ask questions using the same questions box. And at the end of the webinar, time permitting, we will try to answer as many of these as possible. Any questions we don't get around to, we will answer offline and share them with you, along with a copy of the slides from today's webinar. If you are tuning in for the first time, my name's Rssl and I'm the Sterile Manufacturing Lead at Rssl. I work in the commercial team here at Rssl and I work directly with the pharmaceutical microbiology team to support our clients who are involved in both sterile and non-sterile manufacture. For those that have attended our previous webinars, welcome back. And if anybody is interested in those previous webinars, you can go to our website and they're available to download. Rssl can help support you in many aspects of your manufacturing process. And if you want any further information, please just contact me using the contact details you can see on your screen now. So our Meet the Team this month is Rssl. Aaron works in our pharmaceutical microbiology team at Rssl and has worked there for more than 8 years. He is currently the culture laboratory supervisor and the technical go-to person for disinfectant validation, environmental analysis, including running our new moldy TOF, and PET analysis. Welcome, Aaron.
Hi, thank you, Annette.
Hi. So, Aaron, can you give us a brief overview of what your role in Rssl is?
Okay, so my role it can be quite varied. It's on the surface essentially supervising our culture laboratory, but that also is a combination of both supervising the analysts within the laboratory, supervising the analysis that we do. So as you mentioned, disinfection validations, preservative fixity testing, identifications, media QC and so on, will also be responsible for the support operations for the laboratory, the purchasing, health and safety, those kind of aspects.
Okay, and what does a normal week look like for you at Rssl?
So a normal week isn't really a normal week, unfortunately, for for me. So it is incredibly varied week to week. Being a contract lab, it really depends on what our clients need. So some weeks it can be entirely supervisory, so scheduling, managing the staff, and then other weeks it can be full-on just disinfection validations for clients or identification services. So it really does vary week to week, day to day, depending on the needs of the clients that we have on that particular week.
So as we've mentioned, you're heavily involved in running the disinfectant validations for our clients, and disinfection validation is one of the key elements of Annex 1. What are the main challenges with this type of analytical service?
Well, from our perspective, being a contract laboratory, it can be quite complicated to manage these kind of projects because they are bespoke to every single client. So depending on your facility, you'll have different services require different disinfectants, have different organisms that you want to include in the validation. They vary so much. So the actual managing the projects is the most difficult thing from our perspective. For most of our clients, they tend to go the route of having multiple disinfectants, which is usually recommended, multiple of each different types, at multiple Spirocides, multiple general purpose disinfectants, usually at least one alcohol. And generally, from the point of view of the Spirocides, that can usually get quite complicated because trying to find a Spirocide which is not only effective but also well tolerated by the staff is quite important, and from a health and safety point of view, and also your surfaces not damaging them can be quite difficult. So when you're performing a disinfection validation, it is very easy to pick a concentration which works. You're using very small quantities, but also having to compromise so that that concentration is actually usable by the staff in that facility. It can be quite difficult. And then from the perspective of the client's point of view, you have various different services in your facility depending on what type of facility you have. Some of those services can be very easy to validate the disinfection process on, but some can be quite difficult. For example, gloves can be very, very challenging to validate their disinfection because they are a very light, small, flexible surface. A lot of clients request that to be included, but it can be a significant challenge doing that kind of work.
Okay, sounds interesting. And so finally, what do you choose to do when you're not involved in troubleshooting or helping clients with their microbiological requests? What's your downtime look like?
Well, I like to do things that are a little creative in my downtime. I mean, being in a GMP environment, most people that listen to this will be aware, you don't have much opportunity for creativity. It's not really, let's say, frowned upon if you try to be a bit creative in this kind of environment. It's well regulated. So I'd like to do things like more creative, so photography, working in Photoshop, that kind of stuff, where I get to kind of just relax and switch off from the more stringent requirements of the day-to-day job and do something that is just kind of completely different.
Okay, excellent. Well, thanks for your time, Aaron.
No problem. Thanks for having me.
So here at Rssl, we can help, as I mentioned, we can help support you in all aspects of your sterile manufacture process and with Annex 1 support. We can help with environmental monitoring, which includes performing risk assessments for new builds, but also just analysing your routine testing. And as Aaron does, we can identify any of the contaminants with our new mouldy TOF. Aaron can also help, as he's mentioned, with the validation of your disinfectants as part of your cleaning regime. Previously, you'll have heard Jamie, who was involved in the build and certification of our sterility suite. This has been up and running now for, excuse me, almost 2 years, and we can help provide assistance to those looking for sterility assurance. Rssl has extensive experience in providing raw material testing for both the pharma industry and as well as the biopharma industry, and we have really well-equipped labs. We can specialise in those raw materials that need just pharmacopoeia testing, or the more technically challenging ones that you might find in the biopharma industry, as well as being able to perform the suite of microbiological analysis that you might need for your water supply, endotoxin and bioburden. As the sterile industry has grown, we have seen our service for vial and stopper testing grow alongside it, and we can support with these testing both to the pharmacopoeia as well as looking at your integrity testing of these vials and pre-filled syringes. In Annex 1, single-use plastics are becoming more regulated, and it suggests that you know the E&L profile of these. Rssl have a dedicated E&L team to support these activities, as well as looking into any nitrosamine impurities that you might want to detect. And within our microscopy division, we have a range of technical experts that can help with any identification of particulates you may find during your process, or can perform sub-vis testing on your products. And finally, Rssl also has a training team that can help with all your training requirements. This is currently a mixture of online and virtual courses, though we can also put together bespoke in-house courses for you, using a range of consultants to help set up the course to your requirements. And just to mention that Tim is running one of our training courses on the 12th of October on developing a pharmaceutical contamination control strategy. And if you're interested in that, please feel free to contact me. So that's enough about me and Rssl. I'd like to now introduce you to our guest speaker, Dr. Tim Sandle, who I'm sure many of you are aware of, and many of you probably subscribe to his publications. Dr. Tim Sandle has over 25 years' experience of microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards, and he has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both the University of Manchester and UCL. Welcome, Tim. I'll hand over to you.
Okay, thank you very much. Okay, is all slides okay? Can you hear me okay?
Yep,
right,
I
well,
can hear you, Tim.
Brilliant. Okay, right, well, thank you. Welcome everybody to the webinar. So I'm going to talk for about 35-40 minutes, and then we can take questions and answers. So the subject of this webinar is all about spores. And we're going to be having a look at both bacterial and fungal spores. And it's quite an important distinction to look at both of these types of spores, because they present different types of risks, particularly within the clean room environment. And we'll have a look at the specific risks posed, the issues of selecting disinfectants and spirocides, and the some of the reasons why spores can be especially difficult to kill. We'll also have a look at some of the sources of spores and some of the areas of prevention and biocontamination control measures. And we'll also have a look at, as I mentioned, some of the sporicidal agents and some of the factors that affect the efficacy of those agents. But just to begin with, just to kind of set a baseline, I'm just going to go through some very simple microbiological differences. So all bacteria that are living, dividing, are in what's termed the vegetative state. And for the vast majority of bacteria, this is their only state. They're in populations that are either static, growing, or dying. And typical examples are the types of organisms most commonly found on human skin, like species of Staphylococcus and Micrococcus and so on. But there are some bacteria that can also form spores. So generally, this is what we'd find typically within pharmaceutical facilities are going to be species like Bacillus and maybe some Clostridia. Now, these organisms prefer being...
Can I just jump in? Sorry.
Yep.
The GoToWebinar box is over the top of the slide, so...
Oh, okay.
Thank you.
Oh. Let me see if I can get rid of that. Is that better?
Thank you.
Okay. Okay, sorry about that. So they prefer being in the vegetative state, but they do have a survival mechanism, and they produce endospores, and endo means that it comes from within the bacterial cell. And then on the slide, we have some electron micrographs. So the one on the left is of bacterial spores. So these happen to be Bacillus anthracis. So obviously not something you ever want to come across, but typically representative of spores of Bacillus. And on the right-hand side are fungal spores. So fungal spores tend to be associated with filamentous fungi. It's not an exact division because there are some yeast-like fungi that can produce spores, and there are also kind of fungi like Candida albicans that are dimorphic, which can take on yeast and filamentous characteristics. And the fungus shown on the slide there is of penicillium. So with bacterial endospores, saying the reason that bacteria produce endospores is to address unfavourable conditions. So when conditions are not conducive to vegetative cell growth, a portion of the cytoplasm and a copy of the bacterial chromosome undergo a process of dehydration. They become surrounded by a 3, sometimes 4, but typically 3-layered covering. And then this structure is then released from the containing cell and it becomes the endospore. So this is designed to tolerate extreme environmental conditions. So the spore is resistant to desiccation, generally to most typical temperatures experienced, to starvation, and has a degree of resistance to ultraviolet light and even a certain level of gamma radiation, and also to many standard chemical disinfectants. And the spores can remain potentially viable for years. And there's an institute in Sweden that has got some bacillus spores in a bottle of alcohol. And they've been there for over 100 years, I think 110 years. And every year they take out a portion and attempt to grow. And every year so far, they've managed to cultivate the spores, and they've grown to vegetative cells. So it just shows the longevity of organisms in the spore state. It's theoretically indefinite. And then the diagram on the slide is illustrating the process of forming endospores. So we get that portion of the bacterium becoming the spore. So you've got that process going on within the cell. It then produces the middle picture, B, which is the 3-coated layer of the spore. And we'll talk about what those different parts mean. And then that will stay as a spore until conditions are ready for germination, and then the new vegetative cell comes out of the spore structure. And the fascinating thing about this process is that the process can continue to cycle from sporulation to spore back to vegetative cells and then to spores, in theory, indefinitely, unless something happens that kills the vegetative cells or permanently causes the spores not to germinate. So in a sense, it's almost immortality of these types of bacteria. So fungal spores are different to bacterial spores. So fungal spores are microscopic biological particles. And on the slide, we've got a picture of some Aspergillus spores. And fungi produce spores for 2 reasons. So one is that it allows the fungi to reproduce. So this is a very similar purpose to the seeds within the plant world, although the mechanisms are different. And this also helps create the name and the terminology for the spores. So under the picture on the slide are a list of different names which represent different types of spores, which vary according to the types of fungus, their physiological state, and their morphology. Fungi can also produce spores, a special type of spore for another purpose, and these are Chlamydospores. And these are the thick-walled resting spores of fungi. And these are also designed to survive in unfavourable conditions. So it's like a survival mechanism for the spores. There's also another difference in that bacterial spores are static unless they're physically removed. So there'll be a population of spores on a surface, but that's it. They're there until some physical or mechanical activity shifts them. Fungal spores, in contrast, can travel long distances, and the distance traveled depends upon the type of the fungus and the prevailing conditions. And the direction is going to be based partly on the force of dispersal and other vectors at play, so such as air streams or water droplets. And then these dispersed spores, when they reach a surface, a substrate, will then germinate if the conditions are optimal, or for a period of time they can wait and germinate later, or if they're Chlamydospores, they can last for a prolonged period of time. And the other thing about fungal spores is there's a lot more of them, and they're also more likely to germinate than bacterial spores. And the picture on the slide is of a species of Aspergillus, and it's undergoing spore dispersal. So it's like it's a picture that's been captured of that process. So now to why we might find certain microorganisms within clean rooms. So although clean rooms are well-controlled environments, and we might have good control over our incoming materials and lots of other control measures in place, we will still get a level of spores occurring. And whether microorganisms survive within clean room environments will depend upon the types of microorganisms, the environmental conditions, whether there is a survival strategy in place or not, the level of available nutrients, and so on. So there's a number of factors that need to be considered. There's also different types of common cleanroom bacteria. And what's on the slide there is based on a review that I did, which looked at data over a 10-year period. And you can see there that the majority of organisms are bacteria associated with the human skin, but there will be a level of spore formers. The key thing there is about keeping those numbers in control, keeping them low and keeping them out of aseptic areas. And this, comes down to the different designs of clean rooms, different processes, different ways of getting things in and out. And that's kind of reflected in the Annex 1 upgrade, where it particularly emphasizes transfer of materials, and the associated transfer disinfection is probably representing the biggest risk area as you move through the clean room cascade. With fungi, literature suggests that there are certain fungi that are more often associated with pharmaceutical product recalls, and fungi from different sources. So we can have fungi that are more commonly associated with buildings and the environment, which would be the Alternaria, Cladosporium in particular, which is the typical bathroom mold, the kind of black spots that we get, Aspergillus, Penicillium, and so on. And then there are fungi that are more commonly associated with the human skin microbiome, particularly with the release of skin flakes. So here we tend to have Aspergillus and Penicillium, but also Trichophyton, which is associated with things like athlete's foot, and Malcesia, which is the fungus that can cause dandruff around the scalp. It also is interesting from the human microbiome that the largest concentration of fungi is associated with feet, and particularly with the heel, which may again be another background reason why Annex 1 is introducing the clean room socks. But when it comes to specific clean rooms, you're looking globally, then there's going to be variations according to geography, water, and particularly water activity, the amount of organic materials that might be present, some inorganic materials, degrees of pressure, light intensity. All these kind of factors can affect the organism. They can also shift organisms from states that would naturally be viable into either viable but non-culturable, or those bacteria that can form endospores to enter the spore state, or allow fungi to be in an at-rest state. So there's a number of different factors that can mean different clean rooms are slightly different. It also stands that some organisms are more resistant to disinfectants than others. So this is the classic hierarchy from McDonald. And on this list, gram-positive vegetative bacteria are generally the least resistant to disinfectants. Gram-negative bacteria generally are slightly more resistant, and that's because they have the additional outer membrane, which acts as a barrier to the uptake of some disinfectants. Then there are fungal spores, and this is a kind of slightly variable category because most fungal spores are relatively easily killed relative to those higher up on the hierarchy, but there are some fungi that are slightly more difficult. That's then followed by the mycobacteria, who have the waxy cell coat that acts as a barrier to disinfectant entry. The mycobacteria include things like tuberculosis. Then there's parasites, and then bacterial spores very, very high up the list. And this is where sporicidal disinfectants need to come in. And we'll have a look at why bacterial spores are particularly resistant to disinfectants in a minute. And then at the top are prions, the kind of misshaped pieces of protein that have been associated with things like neurodegenerative diseases. So why spores are a concern? Well, in the endospore state, bacteria cannot grow, but because the whole rationale for forming the spore is to survive in adverse conditions, it means that under conditions where most other organisms might be destroyed, if the bacterial spores might be present. And also they're sitting there as ticking away until conditions become favourable, and then germination is possible. And the primary mechanism is having sufficient water available. The other thing is that spores can form relatively quickly. So it only takes typically, for the typical bacillus, about 6 hours for a vegetative cell to become a spore, and a similar length of time for the vegetative cell then to emerge. And then spores are difficult to destroy. And that's another challenge. And the reason that they're difficult to destroy is based on their resistance, which is kind of based on these various layers that form the bacterial spore. And a good analogy is always to think of the spore as like a walnut. You know, it's quite hard to crack. And if you succeed in cracking it, you've then still got further barriers to get through. So spores are resistant to most biocides. And this is because of a kind of impermeability barrier that's formed by polypeptides that make up the spore coat. And simply, it's a barrier that a lot of disinfectants find challenging or impossible to break through. And if then a biocide gets through, then the inner layers, the cortex and the protoplast, function to limit and block the act of chemical diffusion. And also there's a protein that's involved in spore coat formation, which is called superoxide dimutase. And this also helps to protect, even potentially repel, certain chemicals from interfacing with the bacterium. So we said that fungal spores are generally less resistant than bacterial spores, but it still stands that some fungal cells have an innate ability to present a permeability barrier to biocides, and this might just limit time, but in some cases it can stop certain disinfectants from getting through. And also some fungi can secrete enzymes which can interfere with certain disinfectants and inactivate the active ingredient. So this is why sometimes when you're doing disinfectant efficacy testing, that sometimes some of the fungal times, contact times need to be longer, and it may well be because of this enzymatic activity. And this is particularly with chlorine-based disinfectants. And also the sheer numbers, as I mentioned, there are a lot more fungal spores present at any one time, and if they've been dispersed reasonably around the clean room, it can be more challenging to ensure that all of them have been eliminated, and their ability to spread is quite difficult. But also, we shouldn't think of all bacterial spores as equal either. Disinfectant efficacy test data suggests that some bacterial spores are more challenging to kill than others. So Bacillus cereus is harder to kill, is more resistant than Bacillus subtilis, and in turn Bacillus subtilis is more resistant than Clostridium difficile. So there are sometimes differences in the spores. So the spores selected for disinfectant validation should bear some kind of connection to what's being recovered from cleanroom environmental monitoring. Another factor with spores is that some of them tend to be quite sticky, and they have then an ability to bind with surfaces. Obviously, it depends on the surface energy and the hydrophobic or hydrophilic nature of the surface, but some, like Bacillus megaterium and Clostridium perfringens, are quite adept at affixing to surfaces. And this also then increases resistance, because then it becomes harder with the act of wiping to remove spores and to ensure that the biocide is making sufficient contact with the spore. It also stands that some spores, some species of Bacillus, can form a fourth layer as well, so the walnut becomes even harder to crack. And this is with an additional protein called the exosporium. And it's believed that the purpose of this is allows the spore to interact with the surrounding environment, perhaps signaling when it is time to germinate. But this does confer additional resistance, and for example, this is associated with Bacillus cereus, which is probably why Bacillus cereus stands higher up on that hierarchy of resistance. So how do spores end up in facilities? And we looked at some of the geographical differences and some of the variables that might promote certain organisms to survive, and also which might even trigger the conditions for germination. But industry surveys have shown a number of potential sources of spores. So there are the items designed to be introduced into the clean room, or into certainly the first stage of a CNC area. And then there's inner packaging as well. So certain bags have been associated with spores. It's well known that cardboard and paperboard are rife with spores. So one of the reasons why cardboard should never enter a clean room, there should always be a sufficient layer of wrappings to enable items to be transferred through various steps, and certainly this outer kind of material shouldn't be going anywhere near the clean room. Pallets and floor scrubbers have come up fairly high. Trolley wheels, which always stand up as the primary source of spores, and that's probably because, one, trolleys are pushed from room to room, and there's even activities where instead of items being transferred from one trolley to another, trolleys are sometimes pushed between different grades of clean rooms, or even to CNC areas and back again for, say, rubbish disposal. And trolley wheels are very hard to disinfect. You know, if a trolley is large, it's not easy to lift it up and wipe the wheels, for example. Shoes and shoe covers can be problematic, as can certain raw materials as well. Another source that stands up fairly high are those kind of door kickback plates. So these are the kind of stainless steel bits that go on doors that allow them to be pushed or opened with a foot. And they can be quite notorious residues for contamination behind the actual plate. So with raw materials, it's possible to divide raw materials into different categories, and generally drier substances are at risk from spores. But between these different substances, synthetic materials tend to have a relatively low bacterial count, but of course they can contain spores under some circumstances. Dry powders that are derived from materials of natural origin are more likely to carry higher numbers of spore-forming organisms. So this makes testing important and purchasing materials from an approved supplier important as well. I've mentioned the risks of packaging and cardboard, and even for even things coming into a warehouse and unpacking it, laminate materials are generally better anyway, even as outer packaging, because they're easier to clean and less likely to be holding higher numbers of spores. It's important that controls are in place for materials, and particularly when something is transferred from a warehouse to a CNC or into a clean room. For that initial holding stage, it's important that air can circulate, and if packaging is damp, that there's a drying period before those materials are transferred to the next stage, because there are particular risks about cross-contamination from outer packaging to inner packaging. And we may need to have additional controls around materials that are sterile and designed to go into aseptic areas, and they should always be at least triple wrapped in terms of inner packaging. There are other areas where spores can arise, or particular risk factors. So as a general rule, if HEPA filters or air handling systems ever have to be turned off, which might happen for maintenance purposes, then that potential risk of spores being introduced remains much higher. So that's normally followed by some form of Spirocide or disinfection. And obviously never turn HEPA filters or HVAC systems off. And need to be mindful that there's obviously a big push at the moment towards energy saving, which leads to like reducing airflow rates and so on, but that's never done to the degree that could be a source of contamination. You also need to be careful not to make uncontrolled adjustments to temperature and humidity, because they might create conditions that are more favourable for contamination, and make sure that's all covered through change control. It's also important to avoid dust. Dust in any clean room is, again, something that will be problematic for contamination, and also any blocked vents as well. And even in the most highly controlled clean rooms, so if you go to look at the papers published around the NASA Mars clean room projects, where very high-grade clean rooms are being used, there's the microbial assessments show a strong association between dust and spores. So regular servicing of HVAC systems is very important, as is making sure that all sealing voids are intact and there's no risk of anything getting through by the side of the HEPA filter. Spores will also reside in areas where there are cracks, so damage to vinyl, damage to walls, any exposed plasterboard, anything like that needs to be addressed relatively quickly. A further risk area is changing rooms, especially those where outdoor clothing is changed. And it shouldn't automatically be thought that areas like cold rooms are at a lower risk, because most species of bacillus will survive in cold temperatures, and some are even capable of growth. So going back to Bacillus cereus again, it is psychotrophic, so it can continue to grow at 2 to 8 degrees Celsius. Now, people in general don't carry high numbers of spores, but there are risks of transient flora, particularly from outdoor clothing and especially from shoes. So the general good cleanroom practices of effective gowning, using gowns of appropriate quality, frequent glove changes are all important, as is glove spraying. So although glove sprays are not sporicidal, the regular activity of hand disinfection keeps overall microbial levels low. And also, a lot of spores will tend to gravitate to ground level. So practicing best practices when undertaking processing operations. So something falls onto the floor, don't pick it up. That will have to wait for the end of the shift and make sure there's a replacement item. Doing anything to avoid touching areas that might be contamination niches. So there are control measures that can lower the chance of spores being present within an area. And these include effective cleaning, because effective cleaning is required before any disinfection, and that involves using suitable detergents. Disinfecting with appropriate disinfectants and including a sporicide as part of the rotational regime, and ensuring that it's always spraying and wiping, or applying with a mop, or using a pre-saturated disinfectant wipe, because simply applying the disinfectant without mechanical action is not going to be effective. Having good air distribution, having well-designed cleanable surfaces, and focusing on people, and using pre-sterilized multi-pack items, as Annette mentioned earlier, the single-use drive is also quite important. But the central mechanism is the sporicide. So Spirocide is a special type of disinfectant that can kill or inactivate bacterial and fungal spores. And the key variables are concentration and time, as captured in the definition in the United States Pharmacopoeia, Chapter 1072. And because spores are the hardest to kill, by destroying spores, there's also a confidence that vegetative cells are also destroyed. But when approaching the selection of a sporicide, it's important to recognise that the vast majority of disinfectants on the market are not sporicidal. So alcohols, quats, amphoterics, biguanides, all these are not sporicidal. And as was said earlier, many sporicides are relatively unpleasant and come with health and safety concerns. And one of the questions that's sometimes asked is, what's the safest sporicide to use? There isn't really a safe sporicide, because if a sporicide is going to work, it's designed to kill all living things. So there's always going to be a degree of high level of personal protective clothing required, and many facilities use respirators for operators. So we'll have a look at some of the common types of sporicides that can be found within pharmaceutical facilities. So there is chlorine dioxide, which is an oxidizing agent, and it comes as a two-part system, typically sodium chloride and an organic acid, maybe lactic acid or citric acid. And when they come together, it produces active chlorine. And it tends to last longer than ready formulated hypochlorite, but there are still issues of stability and how long the chlorine remains active for. So making up this type of disinfectant solution to use in a bucket, you need to understand how long that remains active for, whether that's 4 hours, 8 hours, 10 hours, whatever, and that needs to be backed up by some form of qualification study. And these days, chlorine dioxide tends to be preferred over hypochlorite, because hypochlorite is effectively bleach and it becomes more challenging for the operators. The other thing about all these chlorine-based ones is that they tend to be corrosive, and that becomes particularly challenging over time to stainless steel. So a post-disinfection wipe is required to remove the chlorine residue. And residues anyway is increasingly becoming a regulatory hot topic, because then there's additional requirement to prove that any remaining residues are not going to interfere with the next disinfectant or detergent that's used in turn, or they might present toxicological risks if they're remaining on pieces of equipment. Another chlorine-based one is hypochlorous acid, which is a little bit of a newer entrant to the market. And this has received alternative publicity in terms of COVID-19, because there's been a lot of discussion around electrolyzed water, and electrolyzed water is a means of generating hypochlorous acid. So this is a slightly more pleasant material to use than chlorine dioxide, and a bit safer, but it might require a slightly longer contact time, and there is still some of the risks associated with stainless steel. Then there is Paracetic Acid, which functions as an oxidizer, penetrating the outer cell membrane and the outer layers of the spore coat, and it is fairly good at sort of rapid deactivation of the cell. But it can be deactivated in terms of variations to pH, and it does produce a very, very, very strong odor that some people find overpowering. It's at its best, it's strong vinegar. At its worst, it's incredibly pungent and does burn the skin if it gets on the operator. Then there's hydrogen peroxide, which is again an oxidizing agent, and it produces these destructive hydroxyl-free radicals that can attack the membrane lipids, DNA, and cellular components. And it has a wide spectrum of activity. It can be an issue in terms of operators and respiration. So using it in closed spaces can be challenging without respirators. And it can kill really well, but equally it's affected by interfering substances. So very, very good quality cleaning is required before the application of hydrogen peroxide, because that can cause issues. And then there are blends of hydrogen peroxide and paracetic acid on the market, which attempt to get a more environmentally friendly approach, because they both break down into water and oxygen, and the synergy of the two is supposed to create quite an effective sporicide, as borne out from disinfectant efficacy studies. But it's important to note that not all sporicides are equal. Need to ensure that we're getting good bacterial and fungal spore kill, and we know that we're able to kill the most challenging organisms that we will find within the particular facility that we're looking at. And we need to have contact times that we can live with as well. So the longer the contact time brings in challenges of drying out, so then you've got to do reapplications, and how long operators can wait and how long a particular process is. So most sporicides are between 5 and 10 minutes, but in some cases they can be quite long, and those length of times can be quite challenging. And we also need to make sure that our variables, the temperature and humidity that we're applying these things to are consistent. And we are able to control time properly. We have sufficient time to apply. We have sufficient time to ensure that the sporicide remains in contact with the surface, and we are able to make reapplications as required. And when we carry out disinfectant efficacy studies in terms of this environmental factor, where most disinfectants are evaluated at 20 degrees Celsius, if we're going to use a disinfectant sporicide within a cold room, then it's quite likely that contact time will increase substantially, and we need to know that disinfectant is efficacious under those conditions. But the ultimate proof of sporicidal control is with environmental monitoring and also the control systems we have in place for getting materials and people into the facility. It's good practice to regularly screen the microorganisms found for environmental monitoring, to trend the isolates, and if atypical organisms or unknown spore formers are being recovered, just go back and re-challenge those disinfectants with that. And also that the environmental monitoring media contains appropriate neutralizer against the sporicide as well as the other rotational disinfectant. So we have a environmental monitoring system that is signaling the true state of control. So whizzing through quite a lot, but we've covered bacterial spores, fungal spores. We've looked at typical cleanroom organisms and why they might survive, some of the resistance mechanisms for both fungal and bacterial spores, some of the reasons why spores get into the facility, and we touched on different sporicidal agents. So thanks very much, and hand back to Annette.
Hi Tim, thanks for that. We've had a few questions come through, so I'll see how many we can get through. First, I've got a question from Kelly that I thought was quite amusing. With feet being such a source of microorganisms, isn't it better to keep cleanroom operatives' personal socks on rather than remove them, causing displacement when they change into their cleanroom socks?
That's a good question, and it's interesting because I was talking with a cleanroom supplier about that. In actual fact, what you're trying to do with cleanroom socks is to purchase very kind of thin socks, and they get pulled over the outdoor socks rather than changing the sock, because it's totally right. The removal of a sock is a big risk. You've got the organisms on the foot. You've got what your hands are touching the sock. Do you have this hand sanitizer close to hand? So it should be an overlay sock rather than a replacement of a sock.
Okay, and I have a question from Amy, and she said, how concerned do we need to be if we are detecting low levels of fungal growth in our grade D clean room on environmental monitoring? It's a room they've recently moved into and they didn't see it in their previous room.
I think in grade D, you will occasionally get some levels of fungi. I think as part of your biocontamination control strategy, what you want to be seeing is none of those same organisms detected in grade C, and certainly nothing going further into any aseptic facility. You also need to obviously ensure that none of that's ending up in intermediate product or whatever the process is going on within that grade D room. And then as part of continuous improvement, obviously try and eliminate the fungi by trying to assess what that source might be, because it might be a structural issue. There might be some fabric damage. There might be something with an air filter, or it might be something being transferred into the room. So low level, wouldn't worry unduly if it's not getting through, but you still seek to eliminate that over time.
Okay. And I have a question from Kim, and she said, what would you recommend to you clean with between cleaning agents and Spirocides, water for injection or a neutral detergent?
I think that given that residues, I think is going to become the next big relative to pharmaceutical microbiology and cleaning kind of hot topic, where it'd be expected to show that after the application there are no residues surviving that could cause interference, product adulteration, inactivation of the next disinfectant and so on, then water's probably a better bet, because you're introducing another factor with a neutral detergent where you've got to show that you get rid of your detergent residues as well. So you need to use a detergent to do pre-cleaning, but after the disinfectant application, if it's not a low residue disinfectant, then water's probably better. Otherwise you get yourself into a conundrum of ever-growing residues.
Okay, and I have a question from Jane, and she says, why do we need to use Spiny spores in disinfectant validation?
Okay, that's an interesting one. So first of all, there's a big difference between the European and US approaches. So the European fungal sporicidal disinfectant standard changed to say that you need to use spiny spores, where the US standards make no difference. So fungal spores can be smooth, what's called reticular, or spiny. And this all relates to what happens when they land on a surface and what kind of angle of the surface is covered by the spore, and how easy that spore is to remove from the surface. So with Aspergillus, it can be in any one of those three states. So it's seen as a greater challenge to use spiny spores because more of the surface is covered and the fungal spore is harder to remove from the surface. So you're kind of doing the worst case challenge. Whereas if you use smooth spores, they wouldn't necessarily be as difficult to kill.
Okay, thank you, Tim. Oh, I think we've run out of time for questions now. Thank you for your time, Tim. Thank you for answering the questions. Very useful subject again for everybody. I'd like to remind you that any questions that we didn't cover in our Q&A, don't worry. I'll forward them on to Tim. And as I mentioned at the start, we'll circulate the answers along with a copy of the slides within the next few days. Our next webinar will be next month on the 27th of October, covering water systems, microbial monitoring and qualification strategy. Don't forget, if you're interested in Tim's course on developing a contamination control strategy that takes place on the 12th of October, please do get in contact. So on behalf of Rssl and the team behind me in the pharmaceutical team, I'd like to thank Tim for continuing to deliver such informative webinars to us all. I thank you all out there for your time for listening today. And as I've mentioned, please don't hesitate to contact me if you have any questions or if you need support with any projects. Rssl are proud to be an organisation that's small enough to care, but big enough to deliver. Thank you and have a good afternoon.
Thank you.